pleased to have Bogumil to take the lead here for the next part and talk about the projects and GP-Write. Bogumil is the Assistant Professor in the Department of Biochemistry at Western University in London, Ontario, and CEO at Designer Microbes. He received his PhD in biology from Western University, where his research was focused on deciphering genetics of nitrogen-fixing root nodule symbiosis. Next, he worked at the Synthetic Biology and Bioenergy Group at the J. Venter Institute in La Jolla, and JCVI, his work focused on developing technologies for cloning of whole, natural, or synthetic chromosomes. I'll leave you to take it from there. You get the gist. Bogumil, thank you.
Thank you so much. And I'm really excited, and I really cannot follow Andrew's talk, but hopefully you guys got really inspired. And it was for me, essentially when I joined Venter Institute in 2010, when I was also really inspired about creating life from scratch, creating synthetic genomes. So I'm gonna give you a brief history how the first synthetic cell was created, and a bit what happened from there, and then I'm gonna go over the roadmap that we discussed in the GP-Write over the last year and a half. And again, this is gonna be something to help us to establish our Canadian roadmap and find what are the challenges that we should answer. So first, how do we create synthetic life today? And briefly, I'm just going to mention those two discoveries that happened at Venter Institute. The first one was the genome transplantation, and this was actually something that got me switching from nitrogen-fixing root nodule symbiosis to synthetic biology. For me, that was one of the most amazing papers, and that was paper when they showed that you can take a genome from one bacteria. So as we have the donor, and then you can actually move the genome into agarose plaque. So you can extract the DNA, store it in this plaque. It has millions of bacteria right now trapped in this agarose plaque, and then you can put it back to the recipient cell. And when you put back to the recipient cells, you can replace the genome of the recipient cells with the donor cells, and eventually you have genome transplantation. And people were doing this with small plasmids. They were moving plasmids from cell to cell. But it wasn't until Carol Artig and her team at Venter Institute worked on this to actually show that it is possible with the whole genome. So nothing was missing. The whole genome could be moved like that. And for me, that was like the beginning of creating life from scratch. Now, this was taken genome from bacteria to bacteria, essentially. But you probably would need a host if you are going to synthesize a new genome. So another discovery that was really important was essentially creating a host so we can park the genomes. Here are three methods how this can be done. And in the first method, essentially what you can do, and this was published in 2010. So in 2007 we have the first genome transplantation. Here is just a review of the methods. How do you put genome into host strain? In times yeast. So in this case, you can take genome. You can take a yeast centromere, original replication as a marker, put inside your microbe, and then you can actually park it into yeast. This microphone is a bit finicky, so that was the simple way. Now, if you cannot move the whole original replications and selection marker in the bacteria, you can isolate the genome in B. You can have your vector that can replicate in yeast. You will need to linearize the genome in a certain way. Now we can do it with CRISPRs. You co-transform it, and then you can move it inside yeast. Yeast is going to recombine that, and you can maintain inside yeast. And in the third one, which was really exciting, you can actually take pieces that are overlapping pieces with small homology, 60 base pairs between large pieces, and you can assemble the bacteria. And and the fact that you can have whole genomes built in such a way, that was kind of revolution. And finally, there was another way that was added later. Actually, when I joined Venter Institute, that to simplify all the three previous methods, you could essentially take and fuse two cells together. So if you pre-insert all the elements that are required, that will allow to replication in yeast, so again, centromere, original replications, and marker, you can put into this bacteria, and you can just fuse them. So it's super simple. So again, cell fusion may become a really huge part of Genome Project-Write. So I wanted to bring this here. Now, having those two discoveries, they were later combined. So essentially, the donor genome, you started with whatever donor genomes, in that case was Mycoplasma mycoides. Based on this genome, synthetic genome was created from pieces. So it was all created from pieces, assembled inside yeast, and then from yeast, again, was isolated in those agarose plugs. Now, this method of isolating high-quality DNA works, but still kind of sucks. So this is one of the steps. If you are thinking of project from GP-Write Canada, finding better way to capture large DNA fragments, whole genomes, will be crucial to get this technology going. And then, from there, you can actually take and boot it up into the same, into similar Mycoplasma strain, Mycoplasma capricolum, and essentially that's how the very first synthetic cell was created that Andrew mentioned. And here it is again. So that was 2010. So now a few years passed. We are getting. It was really, really exciting. That's when I joined Venture Institute. My idea was at that time that since first cell was created, now we're gonna be able to create those cells, really fast and cheap. At least that was my impression. Now, when I went to the lab, it wasn't the case. It was actually, very relatively slow process. Huge group was working on it, and essentially millions of dollars. So it was until 2016 that the next version were published, version 2.0 and 3.0. And those were the genomes actually that I participated, I contributed to working. And again, it was based on the same bacteria, but this time we have biological goal, not just to reproduce the bacteria, but find out what genes are essential. So in the end, the JCVI 3.0 is known as the minimal cell. About half of the genes were removed. So right now we have only 531 kb genome with 473 genes. But once again, it is 6 years later, only a few extra cells created, and it is very specialized group that can really do that. So what else happened since then? So in 2016, there was another paper with the genome transplantation. As I said, this was one of the most exciting things that I kind of experienced, in SynBio. And here again, it was some of the original groups with some people here. Sebastian is here. That again, taking other Mycoplasma strains, isolating DNA, and putting it up into the same recipient cell, Mycoplasma capricolum. So it was advanced. But again, this is 10 years later, and again, it's still just Mycoplasma, which is great advancement. But again, this is one of the fields that definitely need to be advanced. Now, when I was at Venture Institute, and we've been applying for grants to move this to E. coli or other things, and what is really strange, other scientists don't find this very exciting. It was already shown for Mycoplasma, so they think it's trivial for other strains, and the grants were usually not approved. Even they will be completely changing the way we engineer microbes. So this is something for us, how we actually kind of judge our own grants. Some progress was done here, but we definitely need more. And cloning inside these, number of bacteria were cloned. And initially, when we were building the synthetic cells, they were always with different genetic code, and that was a safety feature that the genes would not be probably expressed properly in yeast, and therefore not toxic to yeast. Now, since then, there was a number of strains cloned: E. coli plasma, Cyanobacteria prochlorococcus, Haemophilus influenza. That was one of the very first strains that were actually sequenced that Andrew mentioned. And also, in addition, eukaryotic chromosomes, whole eukaryotic chromosomes from algae, eukaryotic have a megabase. So we know that we can clone all types of DNA in yeast. The transplantation seems to be much bigger problem. Now, the largest thing that we cloned was close to 2 megabases. What is exciting that just a few weeks or last week, there were 2 papers actually in Nature when they took and combined all these chromosomes into 2 or into 1. They were from 2 different groups. One was from Jeff Buche and one from China. And essentially what this paper shows, that 12 megabases molecule can replicate inside yeast. Right? So the assumption is right now we can probably store, close to 10, 12 megabases of bacteria. So let's say if E. coli has 5 megabases, we probably can clone it inside yeast, right? And not just mycoplasma that are about 1 megabases. So that is very exciting. And then just going overall, as you're thinking of where would you fit and how would you engineer other bacteria, this was just summary slide. When I'm showing you that there was like 4 ways how to capture DNA, first inside yeast, that I briefly mentioned. Once inside yeast, you can engineer them in using many different techniques. One of them is CRISPR-Cas9 that you obviously everyone heard. Once you create different molecules in yeast, you can actually start mating them. You can create different molecules in different mating strains, mate them to create final strains, and you have genome transplantation. Now this works really, really well for mycoplasma. Can we have this for most of the bacteria at least in the next 5-10 years? That would be amazing, right? Could we create this process and obviously optimize that further? Now, as we move to GP-Write, so again, this was just, I showed you this one example, just working with prokaryotic cells, how we created the very first simple prokaryotic synthetic genomes. And I know that Leslie later is going to be talking about eukaryotic cells, so I don't want to talk about it. But still, the mycoplasma is still one of the most elegant experiments. When you can build the whole genome, store it, do whatever modification you want to do it, and then bring it back to life. So I think that's what we want to have in the future, that for every cell, we're going to have a way to store the DNA where we can modify it and bring it back to life. Now, for the GP-Write project, we have a number of different categories that we need to improve. Obviously, there's going to be design, synthesis. I put assembly, delivery, installation together because they kind of fit together as we're going to talk about it. And there's also editing. Editing being mostly when we're thinking of CRISPR-Cas9. So you don't write things from scratch, you can edit them later. I highlighted the other 2 in green. I'm thinking they're more dependent on whatever organism you're going to start working with. The design, you can use similar software once you find out, how genes are regulated. That's more or less common to all systems. Synthesis will be common to all systems. But the assembly delivery will be very specific. It's much easier to deliver DNA to mycoplasma versus plant cell, right? But at the same time, they can be using still some of the same techniques. So now, as we kind of keep thinking of goals for this project, and especially tomorrow, we're going to be discussing this. I think it would be kind of nice if we follow what we did for the GP-Write in US. So essentially, we'll put them into 3 different categories. What we call this white category that we want to achieve into maybe 1 to 5 years. And again, those are projects that can be started right away to improve the existing technologies. Then we're going to have something more difficult, the yellow category, maybe 5 to 10 years. Projects that should be possible, but will require much more time, investment, etc. And something that is 10 years from now, essentially unknown or impossible. It's more, we really don't have idea how to make this happen right now. It's all science fiction. But have them, actually write them down and see if we can work towards that. Now, so quickly going over the design. So in the 1 to 5 years, we should develop more collaborative genome design tools. So when we were working on the synthetic cell, minimal synthetic cell that I was part of it, it was actually so hard to keep tracking all the changes. As we were working with multiple labs, like how do we actually physically, when we make all the changes, so it is very easy, some of the softwares are okay, but then later they just kind of everything is looking so messy when everyone just adds the annotation. So we need to create better software that can be shared, and software that handles very large DNA, not just 100 genes or 1,000, whole chromosomes. And then standards, obviously. We will have to have all standards, so we can kind of keep understanding each other. Because, and again, that should be relatively simple. We just have to agree on certain things. And, once we have that, we should be able to design lots of, within 5 years, we should be no problem be able to design simple changes, all kind of mutations, or if we have genomes with repetitive elements, be able to just remove that, clean this up, create things like that. In 5 to 10 years, we want to move it more into chromosome level design. So we can actually predict the structure once we create very large chromosomes. And then we can also actually engineer hundreds of genes at the same time and understand what's going to happen with those hundreds of genes. Not just synthesize them, but design them in such a way that they're going to work in the way we design them to. And more than 10 years, we should be able, those 2 points are kind of together. We should be able to design something that is very complex, essentially whatever functions we will want to create. If mycoplasma, for example, doesn't have cell wall, we want to create cell wall in mycoplasma. We can do this. We have this minimal cell. Whatever we want to do with it. We can create some superior cells. Maybe we want to send them to Mars and they have to be super robust, but at the same time produce food. So we maybe engineer algae to be some kind of superior qualities. And then essentially have this accurate sequence to phenotype in silico whole cell models for all types of cells. So essentially you come and you are engineer. Everyone can be engineer, even in elementary school. And things will be, so that is the goal. Can we create that for all types of cells? So that will be the design. When we go to synthesis, overall oligosynthesis become cheaper and cheaper. So that is really not the biggest problem. Obviously, it can get cheaper, but actually taking them from the small, from 20 to 100 base pairs to actually larger fragments, that's the challenge. So assembling them into slightly larger fragments, kb or more, that's where it's very expensive. So we have to make improvements there. In the 5 to 10 years, as Andrew mentioned, and there are already companies working on this, have new ways to synthesize DNA, have enzymatic DNA synthesis method, make everything longer, faster, and faster synthesis rate. And automation will be key here, right? So we definitely have to invest in automation, microfluidics, and things like that. So we can use, Andrew mentioned, I think, that we're also using this huge volume. You can see it, I actually really like that. If you can see it, they are using way too much. We don't need that much, and it's true, you waste most of it, right? And again, automation can take care of that. So in 10 years or more, routine synthesis, we should be able to routinely synthesize entire chromosomes. Not like even we did at Venter Institute that it takes years, but you design this today and maybe within a few days you have it. A couple million bases. And essentially, decrease the cost of DNA 1,000-fold or more, probably more, to be able to really enable all types of tests. Assembly, delivery, and installation. For assembly, and again, they are close together because when you think about it, once you synthesize the fragments, if you can store them inside yeast, you can assemble them into final chromosomes. Yeast can be used as a molecule to, for fusion, to deliver them to other cells. And then if you understand all the elements, distillation elements of the cell, centromeres and so on, it's going to be all done in one step. So that's why in the first few years, we should really invest into chromosome structure and find, the centromeres, right? Like, what are the centromeres? We just were working recently with algae and we had no idea, how the centromeres are there, what are the sizes, how they replicate, how many per chromosomes. So actually finding this information for all the key species we're going to work, that will be important. Same with order applications, so you can have episomes. And then automate specialized delivery. And again, microfluidics could be really important here. 5 to 10 years, have all kind of specialized host strains. So for example, I keep saying that we can clone a lot of things in yeast. Now, DNA with high GC, it is not possible to clone in yeast unless you add extra order application every 100 kb. Maybe you don't want to do that in your cells. Maybe you don't want to have those interruptions. So, we should invest into developing new hosts that can store high GC, low GC, with lots of repetitive elements. We should have those hosts, or maybe engineer yeast that can do it. But that's something that should be addressed. Methods for delivering of chromosomes into any cell type, that's another thing. And again, centromere, I put, so in the first 5 years we start working on it. Hopefully within the 10 years we have this data already. So centromere origin of replications are identified for the major organism. And 10 years, we have rapid assembly for testing of megabase sized synthetic chromosomes. So megabase, that's minimum, I'm thinking maybe 100 megabase. And something that is kind of crazy, have template independent synthesis of DNA inside destination organism. So the cell is going to be able to read some kind of signal, and based on the signal, it's going to make DNA right into the cell. So you're skipping everything else. How that's going to happen, we have no idea. Here are my few kind of favorite thing, the cell fusion I mentioned a few times. So we had just project, I gave talk in May on GP-Write in Boston, and said that I would like to start engineering cells. And then essentially we start collaboration with Brazilian group when we try to deliver DNA by fusing yeast spiroplast with plant cells. So here we have maize protoplast, and you have yeast that is loaded with DNA right now. And the yeast is kind of, we have express RFP so you can visualize it. And we're gonna be working now, can we move maybe 10 megabases? And whoever's working with plants, they are really hard to work with, even for small fragments. So that's one way. But again, the cell fusion. Now, the yeast is the donor here, but it could be many artificial vesicles that you can design for storing and keeping them, intact till they get delivered. So again, cell fusion is one thing that I think is going to be a big player moving forward. Another one that I really like is conjugation, and conjugation was mentioned as a very old thing. Discover, one of the first things how we discover how DNA is moving, and there are different things coded on DNA. But as we putting, as we kind of researching more of it, you can move DNA from bacteria to algae. Here is algae with silica cell wall and E. coli, and they were moving no problem. We can move to plant cells, we can move to all kind of organisms. And it was shown that recently by ITEA group in Japan that you can move 800 kb. So we're getting close to 1 megabase. Again, so this may be limited in size, but thinking of megabase size molecule, and again, you don't have to isolate the DNA. Once you assemble it inside cell, this is perfect way to keep it intact. And finally, so synthesis is the ideal thing in creating, but sometimes maybe you will just need to edit the cell. So in the next 5 years, enhance all the existing methods, Cas9, talent, zinc fingers, have comprehensive genome scale resources, and optimize by engineering. So we have to, especially for all the species. And in the 5 to 10 years, we would be kind of nice to have spatial and temporal control of epigenetic elements. Also editing tools optimized for all major organisms. So again, we are starting them, in the first few years, we're starting working on them. In the 10 years, we want to have the tools. And more than 10 years, we can just have multiplex genome-wide editing of large chromosomes, hundreds of changes, or thousands at the same time. How could we achieve that without disrupting other things? But again, maybe editing would be so powerful by then. And finally, I'm going to stop here now. As we're talking about Genome Project-Write, we started first with human genome, then we started including all kinds of organisms. And again, one of the major projects in US right now is again focusing on human cell line. But again, we need to involve everyone here, as many people as possible. I personally kind of I want to get there as fast as possible, so we can create organism, really, whatever organism you want. So as we're working with, we should maybe, that's again for discussion tomorrow, create number of different groups. As they're working on viruses or bacteria, yeast, algae, plants, and so on, they're going to be contributing differently to this whole cycle. Right, but we can all really fit into each other, so we find and solve all the problems, how to synthesize DNA and deliver it. So hopefully this project will be much more inclusive moving forward, and it is kind of, but maybe it wasn't so clear. But because people keep saying, oh, I probably don't fit into GPRI because I don't work with human cell lines. And that should never be stopping you to joining this effort. So that's it. Thank you very much.
See if this is working. Are there any questions out there? That was a very thorough road mapping exercise. No questions? Please.
Following the vein, I guess, of this meeting and the technology you're talking about, do you see like Canadian labs in particular that have things that would be related, like technologies that focus on this stuff, or can you, like, I don't know if you have a vision for how Canada's technology environment and what people are doing in this room that contributes to the overall thing?
Well, so I mean, one of the main goals is to actually identify what we have here, right? What we have and who is working on whatever technology. So kind of I would talk about myself. I mentioned this plant, and an algae, and I'm interested in that. So I don't have huge expertise in, mammalian cells, so I'm gonna be working on this, right? And then hopefully I can fit. So if I can store larger fragments of DNA, or if I can improve the conjugation, that we can maybe move 2 megabases to any cells, to algae. Again, this will feed, the other, So again, it's gonna be, today and tomorrow, especially tomorrow, we're gonna go to these exercises to find out what we have. But again, we can lead, certain project and contribute. So I know, sorry, this answer is not precise, but yeah.
Maybe it's a similar question, like what have you seen elsewhere that's already being done that, like I don't know who like of course there's like Jeff Booker and stuff in the US that's happening, but is there any notable names or labs that have some of the stuff covered?
Obviously Venture Institute will be the first one, right? They're always gonna continue pushing the boundaries of creating larger things. They're a company, synthetic genomics, but in Canada, precisely, obviously Vincent is here, the leader of SynBio, and he's pushing. I don't know who is more than Vincent, pushing the SynBio, but I don't know personally who's working on very large fragments in Canada right now. And that is one of the goals of this meeting, right? To find out that that there is need, bring us together. Other than Vincent, maybe you can kind of answer this later, but there is many huge projects like that right now.
Yeah, my name is Mark Sahner. I'm at the University of Ottawa, and I work on issues such as ethics, policy, and regulation. And I'm just curious about how to package this project for ultimately to get funding and so forth. I mean, the way you put it, it is very impressive and fascinating. But it's all about how to make it better, faster, how to improve the platform. You don't really talk in the language of solving problems. And I noticed even Craig Venter, when he was frequently on the news, he's very skilled in saying, so when he's pushed, so why are you doing this? He would say, oh, we will reduce the time it takes to produce a vaccine from half a year to a month. That's something people totally understand. And so I'm just curious, for the 2 days here, I think we need both, but is there a way you see that you can translate this improved platform into sort of the language of problem solving, and also in a sort of responsible, balanced way where people get a sense that you think big about the problems of the world and not just like a particular economic gain, let's say?
Yeah, no, I mean, this is probably the most important thing. I am a young investigator, and I just applied to Genome Canada, and I gave them this big vision. Maybe didn't, precisely address that question, and the grant wasn't funded, right? So yeah, be able to actually, as we developing this technology, and as we kind of have these huge goals, right? We can still, try to, find out, if we optimize the DNA synthesis and it's cheaper, we can still apply to the smaller project, right? We can still apply, and maybe we can test many versions of specific genes. Either it's going to be something with like nitrogen fixation. So here in Ontario, we have, agriculture. Maybe we can contribute to create better microbes in soil, but we may be working only with very few genes. But then, these genes, we may have to test them, thousands or millions of times, all kind of versions. If DNA synthesis is cheaper, so those are kind of small ways. But again, this is probably what we have to figure out tomorrow. What are we going to decide? What are we going to propose? And what people want here in Canada. And probably I will stop now because we have, unless there are more questions.
Yep, no, I think if there's no other questions, and we feel free to bring the questions to Bogumil and others during the cocktail at the end if you have other things that are a little bit more discussion oriented. Well, thank you so much. That was very I did just want to follow up on Mark's question there, because it's a really interesting one, right? For anything that is science-based or engineering-based, you definitely need to be able to express the large thought to communicate with the non-expert community. But at the end of the day, the stuff works because you've paid attention to the actual science and the actual detail. And to a certain extent, speeding it up is the difficult thing. What you apply it to has a lot to do with circumstances or where you are.